Coming to nucleotide chemistry. So, what are nucleotides? Before going to details of nucleotide chemistry, so what is nucleic acid? How it is first separated? So, nucleic acid first isolated by Friedrich Nischner in 1868 from pus cells. So, what are nucleic acid? Nucleic acid nothing but they are polymers of nucleotides. So, many monomeric units of nucleotides they join together to form a polymer of nucleotide. Simply we call it as nucleic acids. So there are two types of nucleic acids, deoxyribonucleic acid and ribonucleic acid. So these DNA and RNA nucleic acids, these are concerned with the storage and transfer of genetic information. So that is the main use of these DNA and RNA. So we call them as nucleic acids. Coming to nucleotides. So we discussed that nucleotides, nucleic acids are the polymers of nucleotides. So what is the components or composition of nucleotides? So these nucleotides have nitrogenous base, they have a pentose sugar, and they have phosphate groups. So this is about the components of the nucleotides. And coming to nitrogenous base, that nitrogenous base may be purine or pyrimidine, and pentose sugar, a 5-carbon sugar, it may be ribose or deoxyribose, and phosphate groups. So these, and the third component is phosphate group. So this phosphate group is esterified to the pentose sugar, and that makes the nucleotides. So nucleoside is nothing but base plus sugar. Nitrogenous base plus sugar is nucleoside, and nucleotide is nitrogenous base plus sugar plus phosphate. So in other way we can say that nucleotide is nucleoside plus phosphate. So this is these are the components or composition of nucleotides. So in this figure you can see clearly that there is base, nitrogenous base joined to this sugar, ribose, and that bond is called as N-glycosidic bond. And because the nitrogen of base is linked to the sugar, ribose, and that's why it is called as N-glycosidic bond. And base plus sugar we call it as nucleoside. And when you see to that, when a phosphate is added, it is called as a nucleotide. And it has one phosphate group, we call it as nucleotide monophosphate, and with two phosphate groups we call it as nucleotide diphosphate, and with three phosphate groups we call it as nucleotide triphosphate. So whatever it may be, base plus sugar and having a phosphate group we call it as a nucleotide. So based on the number of phosphate groups we call it as nucleotide monophosphate, diphosphate or triphosphate. And when you see that sugar at second carbon, if the group is OH group, then we call it as ribose, and if oxygen is removed from that OH group, it results in the formation of sugar called as deoxyribose. So deoxyribose is present in case of DNA and ribose is present in case of RNA. So these nucleotides not only have a role in the transferring the genetic information and storage of genetic information, apart from that, so as a role in the transfer of genetic information, when you see DNA and RNA, apart from that they have some other functions like these nucleotides, they have coenzyme functions. So when you see most of the B complex vitamins, they act as coenzymes, and the B1, when you see B2 riboflavin, it has a coenzyme form called as FAD, and the B3 niacin has two coenzymes, NAD and NAD+. These two are nothing but they are nucleotides. And the other important nucleotide is ATP, the energy currency of the cell, we call it as ATP. That ATP is also a nucleotide, adenosine triphosphate. So it has adenine, ribose and three phosphates esterified to that ribose molecule. So it is called as adenosine triphosphate, is a type of nucleotide. And apart from that, these nucleotides, they also act as regulators and they control the several metabolic processes. So they regulate the metabolisms in our body by acting as allosteric modulators. So these are the important functions of nucleotides apart from their role in the transfer of genetic information. And So what are the bases present in nucleic acids? We have seen that nucleic acids are the polymers of nucleotides. Nucleotides have different components. So one of the component is bases, and those bases are two types. One is purine and the other one is pyrimidine. So purine bases, they have two rings, a 6-member ring and 5-member ring. Fused ring is in case of purines. And there are two types of purines, adenine and guanine. So what is adenine? Adenine is a 6-amino purine and guanine is 2-amino 6-oxypurine. So these two are adenine and guanine are the purines. They are present in both DNA and RNA. And coming to those two are the major purines, and there are some minor purines present in our body. Those are hypoxanthine and xanthine. So these are also purines, but their concentration is very less. And this uric acid is nothing but a type of nucleotide, 2,6,8-trioxypurine we call it as. That is uric acid. That uric acid is formed from the catabolism of purines. Yeah. So that is a type of purine base. Now coming to, there are some purine bases present in food, that is caffeine as a purine base. And coming to another type of nitrogenous bases, pyrimidine bases. So we have seen purines, those purines are adenine and guanine. Coming to another type of nitrogen bases, pyrimidines. So these pyrimidines, they have a single 6-member ring, and there are 3 types of pyrimidines. Those are cytosine, uracil and thymine. So cytosine is present in both DNA and RNA, whereas uracil is present only RNA and thymine is present only in DNA. So what is cytosine? Cytosine is a 2-oxy-4-amino pyrimidine, and uracil is 2,4-dioxypyrimidine, and thymine is 2,4-dioxy-5-methyl-pyrimidine is thymine. So that is about their different groups in these pyrimidines. And there are some modified pyrimidines. So when you add some additional groups to these pyrimidines, so we'll get derived pyrimidines or modified pyrimidines. So those are methyl cytosine. Just you add a methyl group to the cytosine, it becomes methyl cytosine, and other one is dihydrouracil. So these two are the examples for modified pyrimidines. And the second component of nucleotides is sugar. So that is a pentose sugar, and we have 2 types of sugars. That is, we have ribose, deoxyribose. So the only difference between ribose and deoxyribose is at second carbon the oxygen is removed, then it becomes deoxyribose. Deoxyribose is nothing but a type of derived monosaccharide. Right? So this is about the sugars present in the nucleic acids, ribose and deoxyribose. Right? So what is nucleoside? Nucleoside is nothing but it's a base plus sugar. So you can take that adenosine. So adenosine has adenine plus ribose. So adenine is the nitrogenous base and ribose is the sugar. The same way, when you take deoxythymidine, thymine is the nitrogenous base and deoxyribose is the sugar. So these two are called as nucleosides because they don't have phosphate group esterified to the sugar. So that's why we call them as nucleosides. And what is nucleotide? Just you add a phosphate to the nucleoside, then it becomes a nucleotide. So adenosine monophosphate, in short we call it as AMP, and thymidine monophosphate, call it as TMP. So this AMP and TMP are nucleotides because, so when you see the structure, they have nitrogenous base adenine plus sugar, and to that sugar there is an additional group phosphate is attached to the esterified to the ribose of that adenosine. So that we call it as adenosine monophosphate is nothing but a nucleotide. So same way TMP is a thymidine monophosphate having nitrogenous base thymine, ribose and phosphoric acid esterified to the 5th carbon of ribose. Okay. So when you see the bases in the DNA and RNA and their corresponding nucleosides and nucleotides, so when you see adenine and its nucleoside is adenosine, and when it is esterified to a phosphate group, it forms adenosine monophosphate. Generally, that phosphate is esterified to the fifth carbon of the sugar, so that's why it is named as adenosine 5-monophosphate. So in short, we call it as, abbreviation is AMP, and guanine and ribonucleoside is guanosine, and its nucleotide is GMP, guanosine monophosphate. Cytosine, and it's, when it binds with a sugar, it forms cytidine, and with phosphate, that cytidine is converted into cytidine monophosphate. So same for uracil, with sugar we call it as uridine, and uridine with a phosphate group becomes uridine monophosphate. And with the deoxy sugars, adenine with deoxyribose becomes deoxyadenosine, and with a phosphate, that is, it becomes a nucleotide, deoxyribonucleotide, and in case of adenine, it is deoxyadenosine monophosphate. So abbreviation is DAMP. So that D stands for deoxy, and because the oxygen of ribosyl is removed there, and when you see guanine and with the deoxy sugar, it becomes deoxyguanosine, and with phosphate it is deoxyguanosine monophosphate, and DGMP. And coming to cytosine, cytosine with deoxy sugar is deoxycytidine, and with phosphate it is called as deoxycytidine monophosphate. In short, we call it as DCMP. And coming to thymine, thymine with the deoxyribose, we call it as deoxythymidine, and when a phosphate group is esterified to this deoxythymidine, it is converted to deoxythymidine monophosphate, and its abbreviation is DTMP. And coming to structure of ATP, thus energy currency of the cell, we call it as ATP. So that ATP has adenine, nitrogenous base, ribose, and 3 phosphates that are esterified to the ribose 5th carbon. So this is the structure of ATP and nucleotide. Coming to cyclic AMP, this is also a type of nucleotide, but the difference between AMP and cyclic AMP is that when you see ribose, and observe these groups, 3rd carbon and 5th carbon of ribose, those 2 carbons are esterified to the phosphate group. So there is phosphodiester bond in case of cyclic AMP. So because there is cyclic form formed because of involvement of 2 carbons, 3rd carbon and 5th carbon of ribose with the phosphate group, it results in a cyclic compound. That's why it is called as cyclic AMP. So there C stands for cyclic, and that is AMP because it has only 1 phosphate, cyclic AMP. So that cyclic AMP acts as a second messenger, and whatever the nucleic acids that are taken in the diet, so those are converted into corresponding purines and pyrimidines, so with the release of phosphate and sugars by the sequential action of some enzymes. So nucleic acids, they are first reacted upon by the enzymes present in the pancreatic and intestinal secretions. So those are ribonucleases and deoxyribonucleases. They act on the DNA and RNA, and they release the groups from that deoxyribonucleic acid DNA and RNA, and when the phosphate is removed, it results in the formation of nucleotides. Those nucleotides, and again they are acted upon by the enzymes called as nucleotidases, and nucleotidases, when they act on nucleotides, so they release the phosphate that is in the formation of nucleosides. So nucleosides, again they are acted upon by the enzymes called as nucleosidases, so that it releases the free bases and pentose sugars are removed. Right? So this is how whatever the nucleic acids, whatever you take in the diet, they are hydrolyzed by nucleotidase, ribonucleases, deoxyribonucleases, nucleotidases and nucleosidases. Right? So this is the digestion of nucleic acids. And there are some synthetic analogues like mercaptopurine and thioguanine. So they have clinical significance. These two synthetic analogues, they are used in the treatment of cancer. So these two are purine analogues, mercaptopurine and thioguanine. So these two are synthetic preparations. So that's why we call them as synthetic analogs, they have some clinical significance. So till now we have discussed about the definition, what is nucleic acids, what are nucleotides, what is their composition, what are bases present in these nucleotides, what is the type of sugar present in nucleotides, and what is nucleoside, what makes a nucleoside, and how the nucleic acids taken in the diet they are digested, and what are the synthetic analogs and their clinical significance. Thank you.